This is podcast 38. All right, we've been talking about muscle, and we mentioned that calcium is released as a result of the dihydropyridine receptor being voltage gated and then causing a change in shape of the renin receptor and then calcium flooding out of the sarcoplasmic reticulum. But I didn't say how calcium causes actually muscle contraction. So I'm going to talk about this. This is called the sliding filament model. And what we're looking at here is a bundle of myosin fibers and just the myosin head coming off of that bundle of fibers. And then here is a microfilament, an actin fiber, and these are G-actin units on that fiber. So in this model, the sliding filament model, the myosin stays put, the blue stuff stays put, and the pink stuff, red stuff is going to move. So actin moves on myosin. Myosin causes actin to move, not the other way around. And in step one, ATP is going to bind to the myosin, and myosin is needed for unbinding its head. ATP is needed to unbind myosin's head from actin. So if there's no ATP around, at the end of the cycle, you'll see soon, myosin ends up bound to actin. If there's no ATP around, it stays bound to actin, and your muscles are very stiff. This is known as, after death, as rigor mortis, that bodies get very, very stiff, and that's because you've run out of ATP, and your myosin heads are bound to your actin. Later on, when the body starts to break down, you get these protein connections breaking, but soon after death, you get rigor mortis because myosin is bound to actin, and you need ATP in order to relax, not to contract. So then you're going to have hydrolysis of the ATP that binds to this head. It's going to hydrolyze to ADP and P. ADP and P stay stuck even though they're split. They stay stuck to that myosin head, but the action of ATP splitting to ADP and P causes the myosin head to cock forward, to move forward. So from this position to this position happens after you split that ATP, but again, ADP and P stay bound. Then after it's moved its head forward, then it'll bind to a new spot on the actin filament. If you notice here, where it came off is different from where it's going to bind here. Okay? And then it's actually going to move the actin relative to the myosin when phosphate is released. So up here, ADP and P, after ATP is split, stay on the myosin head, and then it binds. Now phosphate is released as a result of that. That's called a power stroke, and that's when myosin actually moves on actin. Okay? Moves actin, causes actin to move, and myosin stays put. Okay? Then ADP is released, and then a new ATP molecule is bound, and you repeat the cycle. So the new ATP molecule is bound. It's split. That causes it. So ADP is released. A new ATP molecule is bound. That causes it to unbind from where it was bound. Then that new ATP is split. It'll cock its head forward. It'll bind to the new spot. Phosphate will be released. It'll cause the myosin to move again in this or the actin to move in this direction relative to the myosin, and these sorts of cycles are what allows muscle to contract. Some more fancy diagrams, other than these cartoons here, in that you actually get the shape of these molecules. So here is just a myosin head with its lever arm there, and when the head gets cocked, in the sense this is after ATP has been bound and it got released from the actin fiber. ATP gets split to ADP and P, but ADP and P stay bound. It cocks its head forward and it looks like this. And then after phosphate is released, it looks like this, relative to the diagram above. So you can see this individual G-actin subunit is the one where it's going to bind, and then that will move to the left side of your screen there relative to where it was in the power stroke. Okay? So, still haven't told you anything about calcium. Calcium is being released from the sarcoplasmic reticulum, and then you're going to get this cycling happening. But how is calcium involved? So you don't have just a naked microfilament of G-actin subunits. The actin is wrapped up in other types of proteins, and those other types of proteins are sensitive to calcium. They include tropomyosin and the troponins. So tropomyosin is this ropey-like thing that is wrapping around the actin filament, and then the troponins include troponin N, T, I, and C, and these are calcium-binding proteins that interact with tropomyosin to cause it to change shape. When tropomyosin is bound to actin without calcium present, it has this sort of shape, and with calcium present, it has this sort of shape. So it looks like the same thing, but what has happened is the tropomyosin has rotated away, exposing myosin binding sites on the actin. And when calcium is gone, it covers those myosin binding sites up. So calcium has to be present in order for myosin to have a place to bind on actin. So the previous slide where we're talking about the power stroke and all that sort of stuff, that only happens when calcium is around. And if calcium isn't around, there's no place for myosin to bind on the actin. And that's the key involvement of calcium. So calcium is flooded into the cytoplasm of the muscle cell. You can have several rounds of binding and unbinding, cycling of the myosin heads on actin, moving actin relative to myosin, and then as calcium is pumped back in to the sarcoplasmic reticulum, you readopt this sort of shape, and now myosin's got nowhere to bind, and so the muscle relaxes. Okay? Troponin C is the thing that is binding calcium, this guy right here, and it undergoes a shape change, and that shape change is transmitted through I and T, and those are bound to tropomyosin, and that's how you go from this shape to this shape. Okay? All right. In a test tube, or in reality, sorry, in an actual muscle cell, but not in model muscle cells that you're studying in a test tube or a dish, it's a lot more complicated. So there are many things that bind calcium in muscle cells in addition to the troponins. These include proteins like caldesmin, calsequestrin, parvalbumin. They all have a different binding constant for calcium. So when calcium floods into the cell, these things will all bind calcium with different affinities, and so the calcium that is left over for troponins to bind calcium can be affected by the concentration of all these other calcium binding proteins. The thing that is determining how fast you can do a relaxation contraction cycling is not how fast you can contract, but how fast you can relax. So how fast can you suck up that calcium that you release and get it back into the sarcoplasmic reticulum? That is what is rate limiting in terms of how fast you can contract. In a fish, if you are studying a fish and how fast they can contract their muscles, you can separate into the contraction phase and the relaxation phase of the muscle contraction cycle. And in the relaxation phase, you can speed that up by expressing more parvalbumin. So you give more cDNA of parvalbumin. That's translated into more protein copies of parvalbumin, and their relaxation speed speeds up. So this is just an illustration of what I'm talking about up here, in that it's complicated. It's not just the troponins. It's also these other calcium binding proteins that determines how fast your muscles are going to work. Also, muscle cells themselves and the actin and myosin myofibril bands inside muscle cells are more complicated too, and there are other proteins that are involved. So if you remember, when we talked about actin filaments, we talked about how they grow and they shrink and how they have polarity. Well, if in your muscle cells, that is not necessarily desirable to have them be really dynamic and change shape a lot, because you want to not have to build your muscle and then use your muscle. You just want to use it. So there's a whole bunch of other accessory proteins that stabilize muscle actin filaments. These include things like CAPZ, titin, and tropomodulin, and nebulin, which is this black one running around. These all function to stabilize this arrangement of myosin on actin, this interaction of myosin on actin. Titin deserves a special mention because it is the biggest protein that has ever been cloned. It is a giant protein that stretches from one unit of muscle contraction to another unit of muscle contraction. This unit is called the sarcomere, and depending on the muscle type, it'll add or subtract amino acids that will add or subtract flexibility to the muscle. And there's lots of different types of titin. So it's a giant protein that stretches all the way across this unit here, and interacts with myosin also. And is very flexible in terms of how flexible your muscles are. So during contraction, this purple line here would get closer to this purple line here, squeeze together like an accordion, and during relaxation they would come further apart. And the interaction is between these heads of myosin in blue here and the actin filaments there. So the myosin is always walking toward the plus end of the actin filaments and away from the negative end. So it's pulling like a tug of war toward this plus end and toward this plus end, and that causes the two purple bands to come closer together during contraction. And then during relaxation, myosin is not attached to actin, and so they can spring back into place. Or there'll be an opposing muscle that will pull them back into place, such as your biceps versus your triceps. Some more about titin. Muscle elasticity that is not accounted for by actin and myosin is accounted for by titin. It's a springy protein. It goes from M line to Z disc. So M line being the middle of the myosin bare zone without any heads, to the Z disc here where the plus end of the actin filaments ends up. It's a giant protein, 27,000 amino acids. Remember I said ryanine receptors were big because they're 6,000 units times 4, so 24,000. This is even bigger. Has a molecular weight of 3 million. myelin, for example, has a molecular weight of 16,000, so it's a giant protein, and you make different versions of it in different muscles, and that adjusts your muscle stiffness. And then nebulin here wraps around the actin filament and helps to stabilize it. And not even shown on here are the troponins and tropomyosin that we talked about before. So it's a very complicated array of proteins in your muscle cells that allows contraction. So most of what is known about muscle has to do with skeletal muscle and contractile, skeletal muscle or cardiac muscle. But there are other kinds of muscle. This is a micrograph of smooth muscle. It doesn't have that regular array that you see in skeletal muscle, less so in cardiac muscle, but you still see regular array in cardiac muscle. Some of the capping and accessory proteins are different in smooth muscle, and contraction is not as fast, and relaxation is also not as fast. But a trade-off is that you can maintain a contracted state with not much energy, only about 10% more energy than the smooth muscle is using in the relaxed state. Okay, here's an H5P.